Right? Now, what is this? Sporopollen. Now, exile in time. Now, just a second. Now, it is the structure of pollen grain. Pollen. It is a white outer layer. Outer layer. Right. Outer layer exile. This exile made with a chemical. That chemical is called as sporopollen. Now, internal pink layer is in time is made with cellulose and pectin. In time, it is the chemical composition. What is the chemical composition of intime? Cellulose and pectin. Right. Exile. This is now. Come to this next one. Now, it is outer covering. That means when you touch the pollen grain, we are touching the outer surface is called as exile, which is made with sporopollen. But be careful. The outer exile at regular interval, it is interrupted, discontinuous. Now it is a discontinuous. It is another discontinuous, where the exile is absent or where the sporopollen is absent. This is the interrupted, discontinuous exile. It is called as germopores. Germopores. What is the germopore? On the surface of pollen grain, some apertures is there. Where the aperture is there, that is name is called a germopore. Aperture is indicate absence of. Aperture is indicate absence of exile. Suppose, for example, this is my body. Some external shirt is there. Internal bunion is there. Sir, that means the skin is covered by the two coverings. Similarly, a vegetative cell is also covered by the two layers, exile and intime. But it is a shirt is nothing exile, the bunion is nothing intime. Suppose the shirt is having some apertures. The aperture, where the aperture is there is no cloud. Similarly, where the aperture is there is no exile. The absence of exile externally indicate as aperture. This aperture is called as germopores. Now this is about the structure of microspore. The microspore is a spherical in shape. The size may be 20 to 50 millimicrons in size. Right? The pollen grain is called as male gametophyte. Why it is called as male gametophyte? Right. It is a young anther, right? It is a mature anther. It is structure of pollen grain, right? In this young anther, right? Theca, microspore, sporogenous cells, epidermis, middle layer, endothelium, tapetum. Just I explain the stuff. But in mature anther, the wall will be ruptured. Due to this, internal pollen grains will be releases to the atmosphere. Anther will be ruptured. Without rupturing of anther, the pollen grains cannot release. Release into the atmosphere, again it is reached with the female reproductive system. That is the surface of the stigma. Some process is there. Right, now it is the rupture. Now hence it is called as mature anther. Now what to do? Before rupturing, what is this total wall? In this wall, outer layer epidermis, endothelium, middle layer. These three layers helps in dehiscence. Dehiscence means it is a Latin word in English, it is a breakdown. That means these three layers must be broken. Right. Due to this, the pollen grains is released. That means function of these three layers. When compared with the tapetum, different function. These three layers performing one function, tapetum is performing another function. These three layers, epidermis, endothelium, middle layer, helps in dehiscence and also give the protection to the pollen. Before dehiscence protection, right? When it is mature, when the anther is mature, these three layers will be there. But now what it is? Tapetum. The tapetum, it is not involved in the days. But what happened? Now what is this internal? Initial stage is microspores. In the middle stage, initial stage sporogenous cells. Initial stage sporogenous cells. Middle stage microspores. Finally pollen grain. All these cells require some food nourishment. What is this? This tapetum provide nourishment to developing pollen grain. Right. When pollen grain will be formed, the tapetum is totally consumed. That means when you see the mature anther, at the time of days, we cannot see the tapetum. The tapetum is already consumed. Hence, the tapetum never involved in daisies. That means the main function of tapetum is providing food or nourishment to developing pollen. In addition to this, now tapetum nourishment. It is also produced enzyme callose enzyme. Now what is this tetroid? Name the tetroid. Microspore tetroid as a cluster together. Now in this tetroid, how many microspores will be released? 4. But this 4 is not free. Now it is a free. Now the tetroid, in between the 4 cells of tetroid, in between the 4 cells of tetroid, some protein is there, that is callose. Callose is a protein helps in together of 4 microspores as a tetrad. Later become 4 microspores. That means the protein callose will be dissolved. The dissolving of callose by callase, which is secreted by tapetum. It is the second function. The next one, ubiscogranules. Ubiscogranules, they form. Outer layer is called as exile. These granules, suppose now it is a pollen grain. Now what is this? Tapetum. From this tapetum, some granules will be there. The granules will be developed. These granules secrete and form exile. That means 3 important functions is performed by tapetum. But according to NCERT book, only one function is there. That is nourishment. Who give the nourishment to whom? Tapetum is provide the nourishment to developing pollen. But one thing, see one thing. Now epidermis, endothelium, middle layer. It is single layer. It is many layer. And now tapetum. Be careful. Each cell of tapetum binucleated. This is the outermost layer is called as epidermis, single layer, single nucleated. Second layer, endothelium, single layer, single nucleated. What is this? Third one, middle layer. The middle layer, how many layers? Many. Multi-layer but unique. But tapetum is a binucleated. One by one, I am going to the back. Just listen. Now, what is this 2? Male gametes. Who is the mother of male gamete? Generative cell. Who forms the generative cell? Microspore. Microspore is formed. Who forms? Sporogenous cell. See? That means diploid sporogenous cells finally become applied male gametes. Okay. Now, what is this structure? Pollen grain. Right. Now male gametes, where it is there? In the pollen grain. And that means the male gametes male gametes is formed in pollen grain or theca. In pollen grain, the pollen grain is present in theca. Hence, this pollen grain is called as male gametophyte. Male gametophyte. Finally, now Now I am going to the completion of this topic. Just finally, just one. It is a triploid sporogenous tetroid 4 microspores it is microspores. Now it is pollen grain. Now pollen grain. Now this is male gamete. Right. All these changes occur theta. Now it is the theta. Now what is this first sporogenous? It is become tetroid. Microspores. Microspore become pollen grain. Name the cell pollen grain. Pollen grain, small cell of pollen grain is called as generative cell. The generative cell within the pollen grain divide and form male gametes. That means finally, this is the two male gametes formed in pollen grain are there in theca. It is a formed in pollen grain which are present in theca. Hence, this pollen grain formation for pollen grain formation for formation of male. Hence, the pollen grain is male gametophyte. Who are called as what? Who are called as what? Pollen grain is called as male gametophyte. Without pollen grain, there is no male gametes. So, in all angiosperms, formation of male gametes require formation of pollen grain. Without pollen grain, there is no male gamete, gametophyte. Who are called as what? Pollen grain is called as male gametophyte. See, now I have drawn the two diagrams. In this here, the anchor is breakdown. You should not say breakdown. Instead of breakdown, you say dehiscence. Now anchor is dehiscence. Now what it is to pollen grains. Right. Here also anchor is dehiscent. And pollen grains are released into the atmosphere. Okay. Suppose 100 plants is there in our surrounding for flowering plants, all flowering plants. Suppose you collect the 100. Among these 100, 60%, in 60%, this pollen grains is a two-cell stage. Two-cell stage at the time of dehiscent. Since 60% angiosperm flowers, their pollen grain containing two-cell stage, this two-cell is generative cell vegetable. But remaining 40 as at the time of dehiscent, this pollen grain containing 3 cells. Among these 3 cells, one is vegetative cell, 2 are the male. This is a little bit difference among the angiosperm. But it is the 2 cell when dehiscent. When it is the reached stigma, right. During this, it is undergoes 3 cells. 2 cell must become 3 cell. Without 3 cell, there is no fertilization. Double fertilization does not occur. Right. Lastly, I am saying differences. It is angiosperm anchor. It is also angiosperm anchor. It is a dehiscent. It is also dehiscent. During dehiscent, the pollen grains are 2 cells. Name the 2 cells. Vegetative cell, generative. It is how much percentage? 60%. Now it is how much percentage? 40% in 40% as the pollen grain is a 3 cell at the time of dehiscent. This is the differences. Now it just took 2 minutes there. Right. Now, right. It is the filament, it is the anther. Anther is maturing and dehiscent pollen grains are released. Now these pollen grains are present in atmosphere. Right. Right. During inspiration, taking of atmospheric air, sometimes we can take the atmosphere with pollen grain because the pollen grain is present in atmosphere. Right. But this causes negative effect in only one cases, that is Parthenium plant. Parthenium plant. Suppose it is a Parthenium plant. It is a Parthenium plant. It is a dehiscent. The pollen grains are releases in the atmosphere. Right. If any person inspire atmospheric air with pollen grain of Parthenium plant, that causes negative effect. Right. Allergy, bronchitis, asthma, chronic respiratory disorder. Chronic respiratory disorder, asthma, bronchitis. This is the three negative effects on the man due to pollen grains of Parthenium. But the same negative, it is not caused by other plants. Is it? It is the negative. But in Western countries, this pollen grains is take as pollen tablets. In some Western countries, this pollen grain, right, due to nutritive values which are present in pollen grain, due to presence of nutritive values in the pollen grain in Western countries, they act as a supplementary food like pollen tablets, pollen syrup. It is a positive effect. Okay. The next one. Right. Pollen viability. What is pollen viability? Now, anther dehiscent pollen grains are present in atmosphere through air, maybe some other agents. This pollen grain is reached to the stigma. Right. This is nothing pollination. We can discuss later. Now the pollen grains are deposited on stigma. After the deposition, they form pollen tube to carry male gametes into ovary or petri. So how it is pollen tube formed, as how the fertilization takes place, we can discuss. But for fertilization, the pollen grain must be deposited on stigma. Before depositation is present in atmosphere. Before atmosphere, it will be dehiscent. Right. Now the pollen grain which are present here, that means after the dehiscent which are present in atmosphere or in water, whatever it may be, somewhere is there, is reach the stigma for further process. This takes place within some duration. That duration is called as pollen viability. The period for which pollen grain remains as alive for the ability germination of the stigma of pistil. If you take some example, you can understand easily. Suppose it is a maize. Alright, this anther belongs to the maize or anther. Now dehiscent is the pollen grains is present. Within half an hour, 30 minutes, it will reach the stigma. Right. Suppose after half an hour, what happened? They become dead. Right. If it is after 1 hour, if it is deposited on the stigma, this is the dead. the continuous processing does not occur. That means some period is required, some period is there from dehiscent anther to deposition. This requires some duration to survival of pollen. Hence, it is called as pollen viability. Once again, definition now catch. The period for which the pollen grain remain as a line for ability of germination of the stigma. After 30 minutes, this pollen grain in rice and maize, they cannot lie. So this duration is called as pollen viability. But in some families, in first year, Fabaceae, Liliaceae, Solanaceae, right? Plant families like Rosaceae, Leguminosae, Solanaceae. In these three families, their pollen grain viability months. Be careful. This is the exceptional. Keep in your mind for NEET purpose and CT purpose. Generally, in theory examination, they will not ask general. But if it is asked, we cannot do it. Right. Now, okay. Some cereals, name the cereals, rice and maize. In these two plants, pollen viability is 30 minutes. In these three families, pollen viability is months. Now come to the pollen bank. Right. Generally, you listen blood bank, eye bank to store the blood. Similarly, these pollen grains we can store. This is called as pollen bank. Right. Some important process is occurring in pollen bank to preserve the pollen grain. That process is called as cryopreservation. Is it? Cryopreservation is the name of a place or process. It is a process. For what purpose? To preserve the pollen grain in pollen bank. Right. In this process, as we use the liquid nitrogen, which containing minus 196 degrees centigrades. Right. Now once again, right, where we can preserve the pollen grain, that is pollen bank. Which process is required to preserve the pollen grain? Cryopreservation. How much temperature? 196 degrees centigrades. Now with this, right, one part, male reproductive system is over. Right. Male reproductive system, generally, male reproductive system, when it will be ending. Suppose the reproductive system function is formation of sperm. Now here in this plant, we should not say the sperm, antherozoid. If the antherozoids is formed, the male reproductive system function is over. Is it? Now, tomorrow we discuss female. Okay, thank you.